In our group, we study the biology of prostate cancer. Why? Well, for one, it's one of the most common diseases on the planet. And it's not necessarily lethal. If tumors are contained within the prostate, there's a good chance of getting effective treatment. However, if tumors spread out to other areas of the body, these metastatic cancers, they're extremely difficult to stop. In the laboratory, we're trying to uncover the process of how cancer develops in the prostate. We want to learn why some cancers get out of the prostate and spread to other organs so that we can identify those tumors at an earlier stage when we have a better chance of successful treatment. And we want to understand what makes the aggressive tumors so aggressive, what gives them the ability to become resistant to our best therapies, so that we can develop new approaches for treatment. You can think of the prostate structurally like a series of branching tubes. The tubes are filled with fluid for reproductive purposes. The lining of the tubes, that's what we're interested in. If you look into one of those tubes, you would see the lining like a ring around the fluid in the middle. There's actually two layers of cells around the middle part, which is called the lumen. The cells that line that lumen are called luminal cells. There's a layer of cells that form an outer ring or base around the inner layer, and these are called basal cells. The basal cells appear smaller and more compact than the luminal cells. Our model of cancer development is that over time, these cells that form the ring structure around these tubes acquire changes to their DNA that give them a growth advantage. And eventually they grow into the middle of the tube, and then they spill out into the surrounding areas. And if they reach the bloodstream, they can travel all over the body and find a new place to live. Now, if you just look at the tumor under a microscope, the cancer cells, they look more like the luminal cells than the basal cells. And so the natural model is that luminal cells probably acquire a series of mutations that give them a growth advantage, and they continue to grow until they form tumors. It seems reasonable, but when someone shows up at the hospital with prostate cancer, we have no way of knowing what happened to create that tumor. So we wanted to test what would happen in an experimental context in the lab. So we worked with doctors and the pathology department to get human cells from patients that were considered normal, not cancerous. And we developed an approach to take cells from the inner luminal layer and cells from the outer basal layer and see what they could do. We put the human cells in an environment where we thought they might be able to grow. We gave them all sorts of goodies to help them grow, put them inside of a mouse. And when we looked, we saw very clear results. The basal cells, unperturbed, they were able to form new tubes, essentially regenerating human prostate tissue in the mouse. These tubes looked just like tubes you would find in a normal human prostate, with a double layer of cells, an inner layer of luminal cells, an outer layer of basal cells. So we learned that basal cells could turn into luminal cells, given the right conditions. We then introduced into these cells what are called oncogenes. They're genes that can induce normal cells to turn into cancer. And when we did this, we found that the basal cells could actually generate prostate cancer. And under a microscope, it looked indistinguishable from what you might see in a man with prostate cancer. So the model we previously described, where prostate cancer probably starts in luminal cells just based on the similarities between normal luminal cells and the cancer cells, that model now needs to be expanded to show that basal cells can turn into luminal cells. And so the basal cells that acquire genetic changes that give them a growth advantage, these oncogenes, they can turn into cancer cells that look like luminal cells. Now, around this time you should be asking, why do we care? Well, we're trying to understand the process of how cancer develops. And now we've identified a very early step. Of course, we wanted to know what happens next. For the basal cells that start the process, do they stick around and continue to produce cancer cells, almost like the stem cell or main source of the tumor? Well, if you want to see what we found and learn how we've studied the next stages in the process of tumor development using this unique experimental approach of converting normal human cells into cancer, check out video number 3.